This will be a recorded lecture on the lymphatic and immune systems of Chapter 20 of your textbook. The lymphatic and immune systems are usually taught together because the immune system utilizes the lymphatic system structures. So we'll start off by talking about how the body prepares itself for invaders and what types of barriers there are to entrance by invaders, and then we'll move into talking about what happens when pathogens and broken down old cells and everything actually get into your system and how your body responds. So the human body's defenses. This is collectively called our immune system, and our immunity prevents the approach of any kind of pathogen. Okay, so a fungus, a bacteria, a protozoan. Okay, so everything in the outside world is denied the entry into our body. If something does get into our body, our immune system prevents the spread of these microbes, and in some cases it does so in a very generic way. All right, so kind of like using a sledgehammer, just getting it all out of the way and not being very specific. Other parts of our immune system allow us to be very specific in different regions and all the way across our body about what response we have to particular microbes. Okay. Our first line of defense are the epithelia. Okay, so epithelial tissues cover things, they line structures. Okay, so this really is our first barrier for entry into our body. Okay, so as we can see, the skin itself provides a lot of barriers, right? The epidermis itself is made of epithelial tissue, okay, and it has a couple specific adaptations to further prevent invasion by pathogens. First of all, we have glands, right? We have sweat glands, we have oil glands, all of these secretions constantly being put out onto the surface of our skin contain different proteins that actually kill or inhibit bacterial growth, okay, sometimes fungal growth, and etc. Okay, so there are bactericidal chemicals, things like lysozymes, which we've heard of a couple times at this point, and antibodies. Furthermore, most of our body is covered by hair, and hair is, among other things, a way that we actually prevent microorganisms from getting close enough to our body to actually invade via the skin. And finally, the structure of our epidermis itself inhibits the invasion of different pathogens, right? So we have multiple layers of skin cells, most of which are dead and just form this relatively impermeable barrier. If something were to get through these most superficial layers of the epidermis, we have the Stratum Spinosum, right? It's the second deepest kind of sub-layer within the epidermis. The Stratum Spinosum has desmosomes, and if you remember, desmosomes are these specialized cell junctions between our cells that essentially inhibit bacteria from getting deeper into our skin. Our skin is not the only epithelial tissue that prevents invasion by bacteria. We have epithelium lining everything that is exposed to the outside world. So for example, in our respiratory system, we have pseudostratified columnar epithelial tissue, which is lining most of our upper respiratory tract. This not only forms this barrier against bacteria, of course, wiggling in between these cells and getting to our blood, but also we have mucus cells, right? We're constantly secreting mucus, as we know, which can, of course, trap pathogens, which is then propelled by the cilia towards the digestive tract, where we would then digest any invaders. Okay, our urinary system, right, system with our kidneys, is cleansed actually by urine. Okay, so urine is kind of flushing out the urethra, washing away physically any bacteria that might be trying to ascend towards the urinary bladder. The reproductive tract is another entryway for bacteria to invade the body, and so the reproductive tract is constantly being flushed, bacteria flushed out of the reproductive tract with glandular secretions, and we'll talk about both of these, the urinary and reproductive systems, later this semester. All right, finally, the digestive tract, as we have spoken about, the digestive system has a lot of different secretions. The stomach produces acid to essentially kill any invaders. Also, there are a variety of structures, as we'll talk about shortly, that specifically prevent invaders from going across this epithelium into our bloodstream. Okay. All epithelial cells can be joined together by tight junctions, and again, this prevents anything, right, any kind of pathogen from getting in between the cells and into our body. Okay, as we can see here, if the tight junctions are not well developed, or if they are compromised for some reason, for example, some chronic inflammation, or this is leaky gut, essentially this allows microbes to get into your blood a lot more easily. So the first thing our body does is try to prevent entry into itself. Okay. However, there are, of course, many instances where invading pathogens actually get into your body, and so we have another line of defense. We constantly patrol, particularly the surfaces of our body, both our skin as well as our digestive system and respiratory system, any entryway into the body, with specialized cells. Okay, down here we can see the cells of the immune system. Okay, we have a stem cell, which can, of course, be differentiated into a lymphoid or myeloid progenitor cell, and these are what give rise to all of the different types of particularly white blood cells. Okay, so we talked about this earlier this semester. This is hematopoiesis. Specifically for this discussion, we are talking about things like the natural killer cells. See, they are close relatives to the lymphocytes. We will be talking about mast cells, right, granulocytes, as well as dendritic cells, which you guys have actually heard about before. Dendritic cells are found in the skin, and again, as we can see, they're derivatives of monocytes, which don't stay in the blood very long. Usually they leave the circulation to become macrophages or specific type of phagocytes, dendritic cells. Okay, so all of these cells are constantly patrolling these surface barriers of the body. Most interestingly to note are the natural killer cells, or the NK cells. These natural killer cells are not only looking for invading bacteria, but they are also checking on our own cells, right? Our cells, particularly in places like our skin and other epithelial barriers, these cells are undergoing rapid mitosis. Okay, the faster that mitosis is occurring, the more likely there will be some kind of a mutation, right? Mutation in the copying of our genome. Okay, so our natural killer cells are not only looking for invading pathogens, they are also looking at our own cells. They want to make sure that if there is a mutation that kind of gets by the cell's internal mechanisms, that we kill them, right? The natural killer cells kill cells that otherwise might become cancer cells, right? A cancer cell is just a normal cell that acquires accumulations or acquires mutations, and as a result can start reproducing uncontrollably, forming tumors, and of course many other side effects. Okay, the natural killer cells are the ones that are actually able to recognize that our own cells are abnormal and physically destroy them. Okay, so here we can see stem cells producing many offspring, many daughter cells. If there is a mutation making a cell, our own cell, slightly abnormal, natural killer cells are actually going to recognize that, secrete some substances, which we're going to look at here in a moment, which literally breaks down our own potentially harmful cells. So natural killer cells recognize, of course, bacteria and foreign invaders, but also they recognize any abnormality in our own cells. Of course, this includes cancer cells, but also anything that is infected by a virus. Okay? Viruses don't have their own technology to replicate themselves, and so they actually kind of hijack our own cells' technology to start making more viruses. And so a cell that is infected with a virus looks very different because it's no longer doing its own job, which of course is for our good, but instead it is working for the virus. And so natural killer cells recognize this abnormality. Okay. So specifically what these natural killer cells are looking for are what are called self-proteins. Right? These are glycoproteins on the surface of our cells. They are unique to us. Okay, so we can recognize our own cellular fingerprint with natural killer cells. Okay, we call these MHC complexes, major histocompatibility complexes, and they are lining pretty much every cell in our body. Okay, we can see off to the right here these markers of self. Here's an epithelial cell with specialized MHCs, MHCs on muscle cells, on leukocytes, on nerve cells. So pretty much every cell in our body has a fingerprint that indicates to natural killer cells that yes, this is us. There is no need to attack this like it is a threat. Okay, to zoom in on that just a little bit, here we have a phospholipid bilayer, so a cell membrane of our own cells. There is a protein inserted into the membrane with a component that acts like a fingerprint. Okay, so the natural killer cells would recognize this particular part and essentially not attack any cell with our unique MHC complexes on the top. Okay, so what we can see here is in a normal cell, there are MHC molecules, right, embedded in the plasma membrane. Natural killer cells will recognize this and will not attack. However, in the absence of this particular configuration, very specific to us and only us, in the absence of that, or if this doesn't look exactly like we think it should, the natural killer cells are going to release chemicals to kill this abnormal cell. Okay, so this would be the case in cancer cells, right? So that would mess up this structure of the MHC molecule, and if something, a bacteria or a fungus or something, does not have this self-MHC at all, natural killer cells respond in the same way. So what exactly are the natural killer cells secreting? How do they kill cells that don't look quite right, whether it's because it's a bacteria or because our own cell is either hijacked or mutated? Okay. What we can see up top here is that another cell that utilizes the same mechanism is going to detect this target cell is not normal, right? We don't want this in our body. And so what it's going to do, zooming in here, is it going to exocytose. So it's going to release chemicals from little granules within the cytoplasm. This includes something called perforins. These perforate or add little holes into the membrane of the target cell, creating pores. All right, and directly after these pores are created, these other molecules called granzymes are going to flow through these channels or these pores, and they are going to communicate with the cell itself, turning on mechanisms coded for in our DNA to stimulate apoptosis. Okay? Apoptosis is programmed cell death, so literally the cell kills itself when it receives these granzyme signals. Okay, so again, when the first line of defense is breached, okay, so when the epithelial tissue, whether it's in the skin or the respiratory system, digestive system, wherever, there is a breach of this defense. Okay? This is going to change the chemical composition of the interstitial fluid, so this fluid that is surrounding all of our cells and our tissues. In response, these damaged cells in this different environment, now that there is an invasion, are going to start releasing different types of chemicals. Okay, they release chemicals called prostaglandins, right, which we'll see are part of the inflammatory response, certain types of communicative proteins, as well as different types of ions. Okay, remember that at this point, when there is tissue damage, hemostasis is going to be triggered. So this is all happening simultaneously. Okay, these chemicals are going to stimulate an activation of the second line of defense. Okay. While hemostasis is occurring, so while we are blocking up that hole to prevent any further blood loss, we have to make sure that any introduced pathogens or proteins don't make their way farther into our body. Okay, and so the first thing that we do after this invasion is something called inflammation. Okay, inflammation involves several different types of cells and chemicals and processes. First of all, we have mast cells. Again, these are relatives of some of the white blood cells we already know about. These are found within the tissues, and they are going to do something called degranulation. So they are going to degranulate. In other words, these granules, which are filled with chemicals that can communicate that, hey, there's an invasion, they are going to be exocytosed, therefore releasing their contents into the surrounding tissues and ultimately getting into the blood. Okay, some of these chemicals are histamine. Okay, you might be aware of this as an inflammatory compound. Okay, other cells that are responsible for this are cells that we know about, like macrophages. Okay, once these chemicals have been released, right, as we can see down here, the mast cells are releasing histamine and other types of chemicals. Macrophages have recognize that there is something that doesn't belong, and they are also going to start releasing chemicals into the local environment, which can ultimately start communicating with the blood and other types of cells within the blood. And specifically, these chemicals are going to lead to the dilation and increased permeability of blood vessels. In other words, more blood is going to flow to the area. Okay, so more blood brings more white blood cells, and as a result, we have more of a defense capability because there are more cells present. Okay. This additional blood flow is going to lead to a warm area. All right, so more blood means more heat, more redness, just because you can see the additional blood through your skin, all of which, of course, tied back to more blood in the area. And also, with more blood pressure in this area, because we have more blood flowing to it, there is going to be a lot more fluid escaping these vessels, okay, and essentially bathing this area of invasion. Okay. Furthermore, this increased permeability allows not only fluid to escape the blood vessels, but also our white blood cells. Okay. These cells respond to chemicals released by the mast cells and the macrophages and any other specialized cell in the area, and they are going to respond to this signal, cytokines, right, something we have heard of before. They're going to respond to these cytokines by literally oozing through pores within the capillary wall, right? So this increasingly more permeable capillary wall. Okay, this process of essentially sniffing out these cytokines and responding to their message is called chemotaxis. Okay, so literally moving to an area based on chemicals within that area. Okay, the process of somewhat oozing out of the capillary wall is called diapedesis, right? Ped for foot, so these are essentially walking out of the capillary wall. Right? Essentially, once they're there, or once they're out of the capillaries and in the site of invasion, these cells are able to complete phagocytosis. So this cell is going to engulf this particular invader and deal with it accordingly, and we'll talk about that in a moment. Phagocytes are the cells that are going to arrive at the scene based on they're following of this chemical trail, so chemotaxis, of the cytokines, of course, and their exit from the capillaries via diapedesis. Ultimately, they arrive in the tissues. They arrive at the scene of invasion, and they are going to engulf and destroy anything that doesn't belong. And so usually this would be a pathogen, but also any kind of cellular debris. Right, so from all the damaged cells that were torn as you got a paper cut, for example, all of those extra components need to be removed and cleaned up from that particular tissue. Okay, there are many different types of phagocytes that are going to arrive at the scene. They all function just a little bit differently, and we'll talk about that in a moment, but in general, they are going to phagocytose in roughly the same way. Right, so we can see here is a bacterium. So this is the invading pathogen. We have this wrapping around of cytoplasm around the bacteria, creating a little vesicle with the bacterium safely inside. Okay, next we have our little cellular stomachs, the lysosomes, right? These are organelles that contain all sorts of different digestive enzymes. Lysosomes are going to fuse, right? They're going to fuse with this vesicle containing the bacterium, and they're going to set their digestive enzymes on breaking down the bacteria. Okay. Now, with these different pieces of the invading bacterium, a couple different things can happen. First of all, any debris that we don't need is going to be released via exocytosis from the cell. Now, certain cells, and we'll talk about this in more detail later, but certain cells can actually take a piece of the bacteria, present it on the surface of the cell, and then show other types of cells, namely the lymphocytes, that this bacteria has invaded the body, and therefore we need to attack it everywhere. Okay, so we'll talk more about that in a moment. But for now, some of the phagocytes that are going to arrive on the scene are things like macrophages, right? The big eaters. Remember that some of these are always within the connective tissue, others are kind of circulating throughout different tissues in the body. These are the big eaters. These guys can eat not just one bacterium, but as many as there are available. Okay. Also, we get the arrival of more cells. Okay, so the arrival within the blood of things like neutrophils. And remember that neutrophils make up about 60% of the white blood cells that you're going to see within your circulating blood. These neutrophils, because they're so abundant, are generally going to be the very first to arrive at the scene. Okay, so the very first ones that arrive escape the blood vessels and start munching on bacteria. Okay, so these neutrophils really do a lot of the cleanup of the damaged tissue. The problem with neutrophils is that they are not big eaters, right? They are not macrophages. They can only eat a single bacterium or a single damaged protein or damaged piece of cell, only one, and then they're going to die. Okay? In fact, the neutrophils arriving at the scene, accumulating, eating a bacteria, and then dying, this gives rise to pus, right? So that gooey substance that indicates that your body is actively fighting an invader. Okay, so the pus really is just dead neutrophils because they cannot eat many bacteria at once. They can eat only one. Also, we have eosinophils and basophils, right? Both of these are other types of granulocytes. These are going to arrive at the scene and specifically phagocytose things that are coated with antibodies. And we'll talk about what produces antibodies and when they're released and whatnot. But in general, these guys are going to eliminate things that have been tagged for destruction already. Okay, and finally, at this point, we have seen many different types of specialized phagocytes throughout the body. Right? Way back in AMP1, we talked about microglia. Right? These are, of course, derivatives of white blood cells as well. They arrive in the central nervous system and they stay in the central nervous system, and they are going to be cleaning up any extra cellular debris and pathogens as well. Okay, we have talked about alveolar macrophages, right? So all the way deep within our respiratory tree, we have macrophages that reside within our alveoli. Within our liver, we have Kupffer cells, which are going to patrol the sinusoid capillaries of the liver. Okay. We have macrophages within our bone marrow. Okay, and these are going to help with the breakdown of used red blood cells after they die, but also can recognize any pathogens that make their way into the bones. We have splenic macrophages. Okay, we'll see here soon that the spleen is also very good at of course, breaking down old red blood cells, but also at filtering fluids from the body to make sure that there aren't any pathogens. Okay, we have intestinal macrophages, okay, so lining the entirety of the intestinal tract, making sure that we phagocytose anything that actually makes it across that epithelial barrier. Okay, and we'll talk quite a lot about the lymph nodes, right, and the specialized cells that reside within the lymph nodes. In addition to phagocytosis, so literally eliminating the pathogens, we have the activation of what is called complement. And complement is a series of reactions. Okay, so something like we've seen before with hemostasis. We activate one protein, which activates another, which activates another, and activates another, and finally at the very end of this pathway, which gets bigger and bigger and bigger as it goes along, we have a series of responses, all of which giving rise to the body's second line of defense. Right? So getting rid of things once they have actually made it past our epithelium. Okay? At the very end here, we have something called opsinization. Okay? Opsinization is when one of these proteins within the pathway, so C3b, is going to actually attach to an invader. Okay, so we actually put little flags on a bacterium, and these little flags are going to essentially call over macrophages or other phagocytes. Okay, so the little flags are very easily detected by these phagocytes, and so bacteria can be recognized and eliminated much more quickly this way. Next, we have cytolysis, so cell lysis, actually like leaking or breaking down. Again, at the end of this pathway, we have proteins that, just like we've seen before with the perforins, they are going to essentially poke holes in the surface of the bacterium. These holes are called membrane attack complexes, or MACs, and these allow the cytoplasm to leak out of the cell. So we're not sending granzymes in to tell the cell to kill itself. Instead, we are allowing it to leak out all over the place so that it essentially, so that it will die. We also have an enhancement of the inflammatory response. Okay, so more inflammation. So we call more mast cells, more basophils to the area. They're going to degranulate, so release the contents of their little cytoplasmic granules, which causes more blood flow to the scene, more delivery of phagocytes, more of these components that can actually break down and eliminate these cells, so generally enhancing the entire response. Complement is also going to increase chemotaxis. Okay, again, sending signals to attract more cells to the scene. Okay, not just flow by in the blood, but actually leave the blood vessels. Another way that the body helps to protect us against invaders that have actually made it past the first line of defense, the epithelia, is a fever. And you wouldn't necessarily think of a fever as being helpful. It kind of makes you uncomfortable and really cold or really hot all of a sudden. But in fact, the fever can actually help your body's immune system. Okay, so a fever is generally anything above 99 degrees Fahrenheit. The way that fevers happen is that your cells, okay, so things like macrophages and other types of white blood cells, release chemicals into the blood called pyrogens. Pyro meaning heat or fire, gen meaning substance. And so these chemicals in your blood are going to talk to the hypothalamus. Okay, the hypothalamus is what is responsible for maintaining homeostasis in general, and so your hypothalamus generally keeps your body at around 98.6 degrees Fahrenheit. Okay, so your hypothalamus is supposed to maintain your temperature within this very narrow range. However, when it detects pyrogens, it actually changes the target set point. So it increases the target set point so that your hypothalamus turns on all of the body's responses to increase heat as if you were cold. Okay, so this can involve things like causing you to shiver, right? Therefore generate more heat. It's going to cause you to not start sweating as soon as you otherwise would to bring your temperature back down. So in general, just bringing up your body's heat. Okay, and so what this can do is potentially inhibit the activity of certain viruses and bacteria. Okay, however, any temperature that can actually kill off a bacteria or substantially inhibit a virus is probably enough to start wrecking your own cells. And so what they think is going on with fevers is that it essentially increases the metabolic rate of the cells within your own body. So it increases how quickly your immune cells are working. So it increases the activity of macrophages and neutrophils and everything trying to get whatever is invading. Okay, also, as we learned in the respiratory system, increased heat leads to more oxygen unloading, and so this further enhances the activity of your body's own tissues. Of course, your fever can't get too high without denaturing your proteins. So by the time you get above 110 to about 112, then your proteins unravel and your body literally starts to kind of cook itself. So the structures and the defenses that we've talked about before are part of what is called the innate immune system or innate immunity. Okay, so we've talked about the physical barriers, right, as part of our first line of defense. These are epithelia, both on the surface of our body and the skin, as well as lining every entry point into our body. We have the patrol by phagocytes, right, so circulating cells within our body, as well as some that are specialized in particular tissues. Immune surveillance, right? We have things like natural killer cells, which literally recognize cells, whether they're our own or foreign cells that don't look quite right. So natural killer cells can actually literally kill cells that aren't quite right. Okay, we also talked about the complement system. Okay, the complement is an activation of a chain reaction of proteins leading to all sorts of different things, ultimately ending with lysed pathogens. Okay, inflammation is a process that starts up right away as soon as tissue is damaged. Okay, so more blood flow, making the area warmer and kind of uncomfortable, but this is actually delivering more white blood cells to the area so that they can clean it out more quickly. Okay, and finally, a fever is supposed to increase the metabolic activity of the cells that are fighting to keep your body healthy. And we didn't talk about one of these, but interferons are actually proteins that they essentially warn other healthy cells that a virus is invading, right? So it's not killing viruses necessarily, it is just making your other healthy cells prepared for viral invasion. Okay, so collectively, all of these different innate immune responses are going to prevent pathogens from really invading your body. It's going to limit their spread, even if they do get past the epithelial barriers, and try to respond accordingly. Okay, all of these responses are really generic. Okay, so they respond exactly the same way, regardless of whether it is an E. coli bacteria or a Streptococcus bacteria. Everything is exactly the same. It's kind of like using a sledgehammer, just get rid of it and don't ask any questions. Okay, so regardless of the type of invading agent. This series of mechanisms, this innate immune system, is called innate because it's present at birth. Okay, we don't have to train this to recognize Streptococcus versus E. coli. It's just there. It's going to act exactly the same way when we're a child versus when we're an adult. Okay, so this is non-specific resistance, kind of like using a sledgehammer. Also, this is focused on just the scene of the attack, so just the damaged physical barrier and the localized invasion. This is not a systemic or body-wide response. Okay, so only local, not systemic. Okay, so if all of these are responses that target anything that is weird, but only in a specific area, how then can we tell the rest of the body that there very well might be an invasion coming? How do we warn the rest of the body that there is an invader that might potentially escape? Okay. We have specialized cells, some of which we've already met, that actually take their digested pathogens, right? So these phagocytes digest the pathogens, they take them, they stick them on the surface of their cells, and they essentially play show and tell. They show T and B lymphocytes that, hey, this is a piece of a bacteria, it's in the body, prepare the rest of the body's tissues to get rid of this particular invader. Okay, so the T and B lymphocytes can actually launch a specific systemic attack on this particular invader. So only the E. coli, not a fungus, not the streptococcus, nothing else, just this particular bacteria. Okay, so this is how the innate immune system can actually trigger a specific systemic response by the adaptive immune system. Okay, so in order for this communication, this warning to be communicated, this requires an antigen presenting cell called an APC. Right? This is just a generic term for any of these phagocytes that can stick pieces of bacteria on their surface. It requires these APCs to be able to actually phagocytose, digest, and present pieces, not the entire thing, but pieces of the invading bacteria. Okay. Furthermore, it does the body no good if these antigen-presenting cells phagocytose something and present it, and they don't actually encounter the lymphocytes. They can't actually warn the T and B cells. So these APCs can be a number of different cells. Some of these are professional. So for example, dendritic cells found within your skin, macrophages, the big eaters, and B lymphocytes also are considered to be professionals, right? That is what they do. Their primary purpose in life. Yet other cells are capable of presenting as well. Things like natural killer cells have this ability, but in general they are focused on other things and will only present antigens or present pieces of bacteria if the opportunity arises. Okay, so as we know, phagocytes engulf the pathogens at the site of infection. They're going to digest it, and they are going to present pieces of it on something that we've seen before, the MHC. Remember that the MHC is what tells particularly natural killer cells that a particular cell is our own. It belongs to us, don't break it down. And so these APCs actually load a section of a bacteria or other invader onto an MHC, presents it on the surface of the cell so that a T cell, in this case, is able to recognize that, hey, the MHC is telling us that this is good, right? The APC itself is our own cell, don't bother it. But whatever is on the surface, right, whatever it is actually presenting, that is what we need to mount an attack against. Okay, so if all of this works out correctly, the T cell, in this case, is going to see the antigen and mount an attack against the antigen, not against our cell. Okay, so what types of molecules might actually be presented in an MHC? Pieces of bacteria or other invader which are distinctive. Okay, so a bacterium, in this case, is covered with all sorts of different types of proteins and carbohydrates. Each of these is considered to be an epitope or an antigenic determinant. So these are molecules that can be recognized by T cells, right, for a variety of T cells, that this is foreign, this does not belong. So they are considered to be immunogenic. Okay. Some antigens have to be somewhat modified before they can produce their own immune response, right? So before they are actually considered to be immunogenic. These molecules are called haptens. Okay, so they're incomplete antigens. The only way that they become immunogenic is to interact with the body's proteins themselves. For example, poison ivy molecules themselves aren't immunogenic. Right? We do not have a response to pure poison ivy molecules. Instead, when these molecules interact with proteins within our skin, they create a different type of protein, and these molecules are immunogenic. Okay, so the only way that we actually get an immune response against them is by interaction with the skin. Okay. As I said, the APCs then present these epitopes, these immunogenic molecules on the surface of our MHC proteins. Okay. Remember that MHC proteins are embedded within the cell membrane, okay, of a dendritic cell, a macrophage, a B cell, whatever we have, and they have one specific component that represents our cells. Right? So this here is highly conserved among all of the MHC proteins within a particular class. Okay, this is going to tell our body that this is our cell, but a T cell or a B cell should recognize whatever is presented here. Okay, we see that this is a peptide binding cleft. This is where we present the epitopes. Okay, now this peptide binding cleft is variable. It is essentially determined by our genes, and we have a different gene for every single potential pathogen invading our body. Okay, so again, genetically determined. The more variable our genes are, the more epitopes we can recognize. Therefore, the more pathogens we can launch an immune response against. Okay. Epitopes can only be bound to these MHCs and presented to lymphocytes if a compatible variable region is produced. And again, these are genetically determined. And so it is very important for us to have as many different MHC genes as possible so that we can recognize many different epitopes. In our genome, the genes for this variable region of the MHC is all contained on chromosome number 6. Right? So as you can see here, collectively, this is called the major histocompatibility complex. And this region of chromosome 6, containing all of these genes, is the most polymorphic section of our genome. Okay? That is, it has the most variability of any other section. Okay? Each of these different MHC genes, of course, codes for a different type of protein, and each of those proteins recognizes a different epitope. Okay. So when choosing a mate, right, or when choosing a mate, it is best to find somebody with a very different MHC group of genes than your own, right? So that your child would then have as many different possibilities, as many different options for recognizing potentially dangerous pathogens as physically possible. Okay, so again, highly advantageous to hybridize, to breed with somebody very different than yourself, someone not, say, your sibling, because you want as many different MHC genes as physically possible. A few years ago, there was a lot of research on this topic, and this Scientific American Mind article came out talking about how a first kiss is our way of essentially assessing the MHC genes in your potential mate. Right? So the article goes on to say that sometimes a first kiss is just the most wonderful thing in the world, like you feel a deep connection with the person that you didn't feel before, but then other times it's absolutely terrible. Right? So what's going on there? You might think you like a person beforehand, but then you kiss them for the first time and, there is no magic. And so what they found out is that the MHC is actually expressed pheromonally in human beings as well as many other organisms, and when you get close enough to somebody to kiss them for the first time, you are actually detecting whether or not their MHC is similar to yours, which of course is not advantageous to your potential offspring, or if they are substantially different than yours, and therefore breeding with this person would produce what is called hybrid vigor, so offspring that is very genetically diverse and therefore able to be resistant to more pathogens. All right, so kind of an interesting article. It's still up online if you want to check it out. Really interesting research. Okay, so again, we have antigen-presenting cells phagocytosing a bacteria, chopping it up, presenting an epitope on an MHC. Okay, so one of these proteins coded for in chromosome 6. Okay. If these APCs never encounter a lymphocyte, the message is lost. We cannot produce a systemic response against this particular epitope. And so the next piece of this puzzle is the APCs actually encountering the lymphocytes, right? Actually passing the message along. Okay. The answer to this, how do we assure that lymphocytes encounter the APCs, is the lymphatic system. Okay, and the lymphatic system is a series of vessels, glands, and organs that actually allow an opportunity for APCs to encounter the lymphocytes. Okay, and so what we can see here is that this is another circulatory pathway. Right, the one we've talked about so far is blood being pumped from the heart into the arterial system, delivering oxygen, nutrients, whatever, to the tissues via capillary beds, deoxygenated blood draining into the venous system, and then returning to the heart. Okay, this system is not within the circulatory pathway. Instead, it is going to essentially suck up anything extra surrounding the capillary beds, send it through a series of filters. These filters are called lymph nodes, or they are called organs like the spleen, which we'll get into. So this extra fluid containing APCs, containing cell fragments, extra proteins that are starting to break down, all of these things are pulled out by phagocytes, right, as well as these lymphocytes within this circulatory system. Okay, ultimately, this filtered fluid is going to be returned to our blood circulation on the venous side of the heart. Okay, so let's talk a little bit more about lymphatic vessels. This network, unidirectional network, begins, right, it's not a cycle, it begins with lymphatic capillaries. Lymphatic capillaries are found in almost every single tissue and organ within your body. For the most part, they are found very closely associated with blood capillaries. Okay, so anywhere that you would have a blood capillary bed, with the exception of things like the brain, you are also going to have lymphatic capillaries. Okay. Essentially, these vessels are substantially more permeable than your blood capillaries. That is, we can have much larger substances passing into the lymphatic vessels that would not otherwise be able to pass into the blood vessels. Right? So, as we can see here, we have these blind-ended lymphatic vessels, and if we look at them a little bit more closely, we see that there are these cells that are not very well joined together. There are these big flaps that form almost like scales, and it leaves these very relatively big gaps in between them. So here we have things like fluid, right? Interstitial fluid between all of the different cells and proteins, able to be sucked up into these lymphatic vessels fairly quickly because it doesn't have to flow through the cellular membranes. Also, big stuff can flow into lymphatic vessels. Things like big proteins, right? So things like albumin can never leave the blood capillary bed because it cannot physically pass over that membrane. That is not a problem with the lymphatic vessels. Instead, we have any big clunky proteins that are kind of floating around in the tissues can easily be taken up into the lymph. Okay. Also, cells, right? Normal continuous capillaries never allow cells to cross over the membranes, right? They're just way too big. And so any cells, such as antigen-presenting cells, can be sucked up into the lymphatic vessels through these big gaps between the lymphatic capillary membrane. Okay, so again, the antigen-presenting cells, which are presenting pieces of antigen, or the epitopes, are going to be sucked up into these vessels and moved along out of this particular area. Now, the lymphatic system does not have a heart or any other kind of pump propelling this fluid along. Okay? So the only way that we actually get lymph to move from, for example, all the way down to the toes and be moved all the way up to return back to the heart is with valves and the skeletal muscle pump. Okay? So just like we saw with the veins, we have these vessels embedded between skeletal muscles, and the more that skeletal muscles move, the more that these vessels are squeezed and then relaxed, and squeezed and then relaxed. And what this is doing is essentially milking up the lymph through through valves. Okay, more pressure down here, pushes open the valve, allowing lymph, or this fluid containing cells and proteins and anything else in the area, is going to be pushed up. And when this area is then allowed to relax, the valve closes, therefore preventing the backflow of lymph back down to the feet. Okay, so this works very similarly to the veins, except for the fact that the veins still have about 10 millimeters of mercury worth of pressure. The lymph has no pressure whatsoever, so you absolutely need skeletal muscle contraction, skeletal muscle movement, in order to propel the lymph closer to the heart. Okay. In the process of getting all the way from the toes back to the heart, these lymphatic vessels are going to pass the lymph through what are called lymph nodes. Okay, so these are small, round, roughly bean-shaped organs, sometimes called your glands, but they're not really secreting anything. Your lymph nodes are populated with specialized white blood cells, specifically lymphocytes, right? So the APCs can actually show the lymphocytes what is invading, and other macrophages just to clean up anything extra. Okay. The principal functional tissue is called white pulp. Okay, and this is actually loose reticular connective tissue populated by these white blood cells. Okay. These lymph nodes are generally clustered in particular areas, specifically around your neck, so the cervical area, right? Someone might tell you have swollen glands. Those are actually larger lymph nodes in your neck. And axillary, so you have lots of lymph nodes around your armpits, right? So if you get a paper cut in your finger, APCs detecting pathogens from that paper cut are going to be milked up these vessels, and they are going to show lymphocytes within your armpit, within the lymph nodes of your axillary region, that this bacteria is invading the body. Okay, also, there is a high density within the inguinal region or the groin area, the vertebral column, okay, as well as mesenteric. So we can see lots of them within our intestines. Note that there are none in the brain. Okay, so there are no lymphatic vessels that we know of in the central nervous system. The lymph nodes themselves, let's zoom in on them just a little bit. As we have seen in other organs, the lymph nodes have a hilum. And a hilum, as we saw, for example, in the lungs, is where vessels enter and exit. In this case, we have blood entering and exiting, and also we have an efferent lymphatic vessel. That is, lymph has been filtered by the lymph node and is now exiting the lymph node to be propelled again back towards the heart. Okay. Note that there are many more afferent vessels than there are efferent vessels. That is, there are many more lymphatic vessels that are bringing lymph to the lymph node, as opposed to the single vessel taking lymph from the lymph node. And so what this does is it allows a lot of lymph to come in, but only a little bit to leave, causing or almost forcing that lymph to stay within the lymph node, allowing macrophages to clean out any damaged proteins, any extracellular debris, allowing time for the APCs to actually present their pathogen or their epitope on the surface of their MHC to the lymphocytes. Okay, so it is here, while the lymph is just hanging out within the lymph node, just kind of waiting in line for it to be able to exit via the single efferent lymphatic vessel, what we have is the APCs activating the lymphocytes. Okay, this actually activates the adaptive immune system, right? So a very specific yet systemic response. It has memory, right? So it remembers something that you've already been exposed to. And it allows proliferation of the lymphocytes. That is, if there is an invasion, we need to make more and more and more lymphocytes so that we can actually respond to the invaders. And so when we have swollen lymph nodes or quote-unquote swollen glands, this means that the cells within your lymph nodes are multiplying, therefore causing the node to be much larger, right? So that is when your tonsils or your lymph nodes or any other structures here get larger. Okay, so on its way back to the heart, the lymph is filtered not just by one lymph node, but by tons of lymph nodes. Okay, so it's filtered down here in the inguinal region, it's filtered again up here, it's filtered pretty much from anywhere that it is coming from, and finally is then dumped back into the blood circulation, right? Nice and clean without any invaders. Okay. The lymphatic system, for reasons unbeknownst to anybody that I know of, is not symmetrical. That is, lymph from 3/4 of the body, right? The left arm, both legs, the mesenteries, all of this drains into the left subclavian vein. Whereas the right arm, right, half of the thoracic cavity and half of the head, this other about quarter is going to flow into the right subclavian vein. Right, so most of the body drains back into the left, not the right. The lymph nodes are by far not the only lymphatic tissues that you have in your body. You have quite a few others that are their own organs or associated with other organs that we have already learned about. First of all, we have an organ called the spleen. Right, the spleen we have heard about before because it is important in essentially filtering broken down red blood cells out of the blood so that we can recycle as much as physically possible. The spleen is located in the upper left quadrant, that is right to the lateral side of the stomach. It also has a hilum, right? So blood enters and exits as well as lymph can enter and exit as well. The spleen has kind of a dual function. Of course, we know already that it filters out used red blood cells. Okay, but now we also know that it contains white pulp as well. So the same kind of stuff that is found within the lymph nodes. Okay, this white pulp tissue is, of course, populated with lymphocytes and macrophages, and APCs are capable of presenting their epitopes or their pathogens to the lymphocytes in this particular organ. Okay, the white pulp, which you can see over here, is surrounded on all sides by the red pulp. Okay, and as we know, this destroys worn-out red blood cells. As a fetus, this is where a good portion of our red blood cells are made, and also it serves as a blood reservoir. Right? So blood can kind of hang out there, and then if we need additional blood circulation, we can mobilize this extra blood. Other structures that we are somewhat familiar with, the tonsils, right? This is a ring of the lymphatic tissue around the pharynx, right? So anything that we take into our mouth or take into our nose has to pass by this ring of tonsils, right? So again, just loose reticular connective tissue that is populated with lymphocytes, which can, of course, recognize any invaders on their way into your body. And in the same way that lymph nodes swell up, the tonsils can swell up as well whenever the lymphocytes are completing mitosis, right? So if you are actively fighting against something, you are going to have swollen tonsils, swollen lymph nodes. Certain regions of the intestines, particularly the ileum, the last part of the small intestine, is populated with what are called Peyer's patches, right? And as we know, Peyer's patches are right under the epithelium, as we can see over here. So this is epithelium. Here we can see the intestinal crypt secreting things like bicarbonate-rich fluid. Here is a villus, right, to increase the surface area for secretion as well as absorption into the blood. Okay, but in the ileum alone, we have Peyer's patches, and as we can see, any antigens, any bacteria, fungus, whatever that has gotten into your body, maybe escaped from the cecum via the ileocecal valve. We don't want that to go all the way up our intestines, and so these these antigens are pulled into the Peyer's patch, and we can see that there are T cells as well as macrophages that are present here, and so they can immediately phagocytose this antigen. And of course they can milk it or get milked via these lymphatic vessels all the way up to a lymph node. And we also have T cells which can launch an immediate adaptive immune response. Okay, we have our vermiform appendix. This is again populated with T cells and B cells, and mucosal associated lymphatic tissue, or MALT. Okay, so this is lymphatic tissue that is immediately deep to the epithelium, right? So if anything does get through those junctions, it is met immediately with lymphatic tissue. Okay, we've heard of a couple of these before. We have GALT, gut-associated lymphatic tissue, and so these little patches of reticular connective tissue that is populated with T cells, B cells, and macrophages. This is lining your entire gut. And in fact, your digestive system really does house about 70% of your immune function, right? We purposely take foreign substances into our body. We send it through this tube, many feet long, and we keep it in our body sometimes for a couple days. And so we need to make sure that nothing utilizes that opportunity to get across the epithelium and into our body. So GALT is very important to keeping us healthy. And so keeping our epithelium intact with our gut flora is very important to our immune system, because this is where most of our immune function is actually housed. We also have these patches of lymphatic tissues immediately deep to our respiratory epithelium. Right, so this is called BALT. Bronchus-associated lymphatic tissue works largely the same way. We purposely take foreign air into our body and therefore make ourselves vulnerable to invasion. Another lymphatic tissue is called the thymus. All right, the thymus is not, I repeat, is not the thyroid gland. All right, so here is the butterfly-shaped thyroid gland. It is right inferior to your larynx. Okay, this is secreting things like thyroid hormone, calcitonin. So this is an endocrine gland, right, secreting hormones. The thymus, on the other hand, is lymphatic in nature. That is, it is important, specifically in the development of T cells. Okay, T cells have a little bit more of a method for maturing. Right, we'll learn about this more later. But T cells need to essentially learn to recognize us, so our cells versus non-self. Okay, and this training process is going to occur within the thymus. Right, so the reason they're even called T cells is because they finish their development in the thymus. Okay, so your thymus is very important during your childhood through the time that you hit puberty. Essentially training your T cells, helping them to learn just different types of things that you will be exposed to throughout your life. So after puberty, your thymus starts to atrophy. That is, it starts to break down and be converted into fat. So it's no longer functional as a thymus anymore, but it does store some energy. When you guys dissected hearts in the animals, the fat on the top of the heart, so on the superior surface of the heart and the blood vessels, in part used to be the thymus. So a couple things can go wrong with the lymphatic system and can actually be very detrimental. Any kind of obstruction of lymphatic vessels leads to edema. Remember that your capillaries, your blood capillaries, can only reabsorb about 99% of the fluid that they release every single day. And so that extra 1% needs to be kind of sucked up into those very permeable lymphatic vessels and milked back to the heart. If there is a blockage in one of these vessels, it leads to tissue swelling. We can no longer take up all of that extra fluid that was released by the blood capillaries. It just stays within our tissues. Okay. In certain circumstances, this blockage can be because of a parasitic invasion or a parasitic infection. The most extreme example of this is called elephantiasis. This is a mosquito-borne protozoan that is found mostly in tropical areas. So this map over here on the right shows you the locations where elephantiasis can occur. A mosquito essentially bites you, releasing this protozoan within your blood. This protozoan breeds, and its offspring actually block your lymph nodes or your lymph vessels. Okay, and so what that means is that anything distal to that site, okay, so for example, here is the mosquito bite up here on the right, all of the fluid that would otherwise be milked along these lymphatic vessels, pass through lymph nodes, and ultimately go back into your blood circulation, gets blocked, right? So anything distal to that blockage. And so what that does is it leaves the fluid within your hand, or whatever is distal to the blockage, leading to things like this. So this is elephantiasis. You can see, of course, much larger than a normal organ. Also, over time, you can get this kind of elephant-like skin, right, without proper circulation. You can't cornify or harden your skin appropriately, and so it leads to excessive cornification. And of course, anywhere that lymph vessels are blocked, even in places like your face, can lead to elephantiasis. And of course, this is the most extreme example, but there are many other ways of blocking or disturbing lymph from flowing. Okay? For example, any kind of surgery that removes substantial tissue, such as a mastectomy, this removes with it the lymph vessels. So without these lymph vessels, fluid is going to accumulate in that place, right? So this is very common, things like mastectomies. Furthermore, again, much less extreme, but still equally troublesome in particular individuals, is edema can be caused by limited use of your skeletal muscles. Remember that there is no pump to actually propel lymph along your vessels and bring it back to your heart. Instead, we need to use our muscles. And so if you are recovering from surgery, right, or you are paralyzed, right, sedentary lifestyle, any reason that you wouldn't be using your skeletal muscles might lead to extra swelling in those extremities. Okay, and so if you've ever been on a plane sitting there for an overseas flight or something, you might notice that your ankles are pretty stiff and maybe a little bit puffy after sitting there for so long. I guess the only reason they are that way is because you haven't been moving. And of course, you are leaving your feet down, so against gravity. Your lymph has to be pumped without the help of your skeletal muscles in this sedentary situation. All right, so one way to kind of help your lymph along and reduce swelling in your extremities is to essentially work with gravity. So stick your feet in the air and help the lymph flow down towards the heart instead of up towards the heart. Right, so this is a common practice in yoga. At the very end of your practice, you might do something like a waterfall, right? So sticking your feet in the air and maybe moving your ankles around a bit, just trying to squeeze any of that extra lymph and fluid back into your bloodstream. Okay, so to summarize the lymphatic system is important in conducting excess fluid. Right, so the extra 1% not reabsorbed by the blood capillaries is going to be absorbed by the lymphatic capillaries. It's not merely enough to reabsorb this fluid. Instead, we actually filter this fluid, now called the lymph. All right, so there's filtration and phagocytosis. Any extracellular debris or pathogens can be eliminated from the body. All right, also, lymphatic system structures are populated with lymphocytes and other types of specialized white blood cells. These are going to help with the immune system of the body, particularly when an APC in the lymph presents an epitope to a lymphocyte. That is going to trigger an adaptive immune response, and that is the subject of the next lesson in lecture.